Good day, this is the Professor, CBMD PhD, and today's Monday, March 13th, 2023. It is 7:36 and 58 seconds, p.m. Eastern Standard Time, and today's research, Bacteriophages. So let's hop to it here. A quick new way to screen virus proteins for antibiotic properties. Bacteriophages can hold the key to combating antibiotic resistance. Working with them just got a lot easier. Date March 13th, 2023. Source: DOE, Lawrence Berkeley National Laboratory. The scientists say a whole new world of antibiotics is waiting inside the viruses that infect bacteria. Scientists are making it easier to study them. One major challenge standing in their way is this difficulty of studying individual bacteriophage phage proteins and determining precisely how the virus smells these tools that kill their host, bacteria. The researchers developed a high-throughput genetic screening approach that can identify the part of the bacterial cell targeted by a potent type of phage weapon called single-gene lysis proteins. With rising antibiotic resistance, we urgently need antibiotic alternatives, some of the smallest phages that we know of code for single-gene lysis proteins, also known as protein antibiotics, to inhibit key components of bacterial cell wall production that, when disrupted, consistently kill the cell. As there appears to be at least one type of phage for every known strain of bacteria, they are thought to be the most abundant biological entities on Earth. In fact, there are an estimated 10 to the power 31 phage particles on the planet right now, or the equivalent of 1 trillion phages for every grain of sand. Each of these phages evolve alongside their chosen host strain, allowing them to counter bacterial resistant traits as they arise with improved biological weaponry. And this massive abundance, specificity, and efficacy means that there are plenty around to study, and that we should theoretically be able to use phages to control any harmful microbes. As phages are harmless to non-biological cells, another reason they are so appealing as medicines and biocontrol tools. The problem arises when trying to isolate a single phage from the environment and determine which microbe it targets and how. The scientists are unable to assess phage bacteria battles based on genomic sequence alone, or study them in action because many bacteria can't be cultured in a laboratory. And even if they could, there is an inherent catch-22 of needing to know ahead of time which bacteria to culture in order to study the phages that infect and kill them. As to sidestep these obstacles and identify the cellular targets of single-gene lysis proteins, the researchers used a technology the team previously invented called dual barcoded shotgun expression library sequencing. Dual barcode shotgun expression library sequencing allows scientists to employ a coded library of DNA fragments to investigate how unknown genes function and can be applied to complicated environmental samples that contain the DNA of many organisms. No culturing needed. In this study, the researchers used single-gene lysis proteins from 6 phages that infect different bacteria and identified the part of the bacterial cell wall or supporting molecules that each single-gene lysis attacks. As this work showed, that single lysis proteins target pathways for cell wall building that arose very early in the evolutionary history of bacteria that are still used by nearly all bacteria, including pathogenic bacteria. And since the single-gene lysis proteins attack such fundamental and ubiquitous targets, they can kill bacteria other than the phage's target strain, confirming they have great potential as antibiotics. The phages are extraordinary innovators when it comes to destroying bacteria. They are really excited to uncover novel bacterial pathogen targeting mechanisms that could be leveraged into therapies. Now that the team has evaluated the dual barcoded shotgun expression library sequence for tackling this question, they can apply it to the thousands of single-gene lysis-producing phages awaiting characterization in environmental samples that the team has collected from the ocean, soils, and even the human gut. The inspiration for the next breakthrough medicine could be in there waiting. And this is the Professor of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.